Good morning to everyone. The title of my lecture is Mushroom and Spawn Production. At the end of this lecture, you're expected to be able to identify the materials needed in mushroom and spawn production and explain the process involved in the production of mushroom and spawn. The Philippines remains to be an agricultural country that produces different crops such as rice, banana, sugarcane, coconut, fruit crops, and many others. During production, various agricultural waste are generated such as rice straw, sugarcane bagasse, coconut husk, banana leaves, corn leaves, stalks, and many others. These are potential substrate for growing different species of mushrooms. The Central Luzon, where the Central Luzon State University is located, is a rice producing region, so that large volume of rice straw is generated. However, rice straw remain underutilized despite its potential use in agriculture. Some are used as animal feeds and mulching materials. Burning of rice straw in the field is not only common in the Philippines, but also in other Southeast Asian countries. It is not only detrimental to the environment, but also to human health. It emits carbon monoxide, carbon dioxide, aromatic hydrocarbon, and sulfur dioxide. For this reason, the Central Luzon State University developed the zero rice waste technology. As what I've mentioned, Central Luzon is the rice granary of the Philippines, so that during rice harvest season, a lot of rice straw and rice bran are generated. The rice straw can be used as a substrate for growing different species of mushroom. Initially, in mushroom production 1, the rice straw can be used in growing the paddy straw mushroom, or scientifically called Bulbariella bulbacea. After producing the first mushroom species, waste is produced in the form of mushroom spent 1, which can be formulated and used as a substrate for growing another species of mushroom, like, for example, the pan-shaped mushroom or Pleurotus florida, or Ganoderma lucidum, or the jello fungi or auricularia. After growing the second species of mushroom, another waste is generated, which is called mushroom spent 2. The mushroom spent 2 can be used as substrate for organic fertilizer for crop production, or can be formulated to be used as animal feeds. After crop production and animal production, crop refuse and animal dung, the mushroom spent 2 can be used as a substrate for organic fertilizer for crop production, or can be formulated to be used as animal feeds. After crop production and animal production, crop refuse and animal dung are produced, which can be used in fertilizing the rice plant. So in this scheme, you will notice that there is complete utilization of the rice straw. There is nutrient cycling and energy flow. Traditionally, mushrooms are cultivated using sawdust. However, the supply of sawdust in the Philippines is becoming limited. In view of this, the Center for Tropical Mushroom Research and Development developed the rice straw-based substrate formulation, which can be used in growing different species of wood-rotting mushroom. The different steps in the production of spawn and mushroom using this technology includes soaking, composting, chopping, substrate formulation, bagging, pasteurization, cooling, inoculation, incubation, growing, and harvesting. In the next slides, I will discuss the details of each step. The first step in the production of spawn is soaking. Soak the rice straw in a water tank for 3 days. There are two reasons why we soak the rice straw in water. Number one is to absorb moisture, and number two is to soften the rice straw in preparation for composting. After 3 days of soaking, drain the water in the tank. Then this is followed by composting. Hold the rice straw from the tank and transfer to the composting area. Cover the pile of rice straw with a plastic sheet or vinyl sheet in order to stimulate the growth of the natural decomposers, which will initiate the decomposition process. Compost the pile for 7 days with turning every 2 days. On the last day of composting, aerate the compost by turning it with a spading fork. The third step is chopping. Chop the rice straw approximately 1 to 2 inches long. Then this is followed by the formulation of the substrate. Formulate the substrate by mixing 7 parts composted rice straw and 3 parts sawdust. Be sure to mix the rice straw and sawdust thoroughly. Check the moisture content of the formulated substrate by getting a handful and then squeeze it. If there is no water coming out in between the fingers, the moisture content is approximately 65%. However, if water comes out in between the fingers, the water is more than 65%. Air dry the substrate until the moisture content is 65%. The next step is bagging. Bag the substrate in a 6 by 12 inches heat-resistant polypropylene plastic bag, or we can call this as PP bag. To easily compact the bags, you can use a metal molder. Put the PVC neck with cotton or garment waste at the open part end of the bag. Cover it with a clean paper and put rubber band. This is the sample of bag substrate which is ready for pasteurization. Pasteurization is being conducted to kill the microorganisms present in the substrate. Pasteurize the bag substrate in a pasteurization chamber with a temperature of 60 to 80 degrees Celsius for 8 hours. In the CLSU Mushroom Center, we use a pasteurizer with a capacity of 500 bags. After this process, allow the bags to cool down overnight and transfer to the inoculation room. Then after 24 hours of cooling down, inoculate the pasteurized fruiting bag with grain spawn of the desired mushroom species. Prior to inoculation, press the grain spawn to separate the grains from each other, and each grain contains the mycelia of the mushroom organism. Then this is followed by incubation. Incubate the inoculated bugs at room temperature to allow the spread of the mycelia. It will take about 3 to 4 weeks before the bug is totally ramified with mycelia. And this is the appearance of totally ramified fruiting bag. When the fruiting bags are fully ramified, the next step is growing. Hang the spawn bags inside the growing house. and open the fruiting bag by cutting one end of the plastic. And this is the appearance of open fruiting bag. Using a mister or a sprayer, water the open end of the bags with clean water at least 3 times a day. One in the morning, noon, and afternoon in order to prevent drying of the bags. After 2 to 3 days from opening, you will note that small mushrooms are coming out. Then after another day, this will be the appearance, and another day, this is the appearance of the mushroom. So these are matured mushrooms that are ready for harvesting. During harvesting, pull the mushroom with your bare hands and do not use knife or scissor. After the first harvest, remove the remnants of harvested mushrooms by gradually scraping until new substrate is seen in the bag. Water 3 times a day, and then clean the house after harvest. After 7 to 10 days, another batch of mushroom will start to come out. Thank you very much for listening.